Cool. So just talked about DNA replication, how that process works. That is all going to happen before the cell divides. Okay, so now let's jump in and talk about what's going on during a cell's life cycle. The cell cycle, if you would, right? So we're going to talk first about interphase, and then we'll jump into mitosis, the actual process of dividing those cells. All right, here we go. So cell cycle is kind of like the life cycle of a cell. Okay, so they go through periods of growth and development, and then they divide if they're given the right signals, right? So here, let's talk about what's going on for most of the cell's life cycle is going to be sent in this section right here. These three phases, which are called, altogether we call them interphase. There's 3 different sections of interphase, okay? During this process of interphase, the cell is not dividing. It's doing its normal cell stuff, okay? If the correct signals are given, the cell will then enter into the process of mitosis to make a copy of itself and then divide up in the process of cytokinesis, okay? So this process, mitosis, the mitotic phases, results in 2 identical copies of a cell, but most of a cell's lifetime is going to be spent in this interphase. So let's see what's going on there in interphase. So the first phase of interphase, what we call G1 or growth 1 phase, is the cell has just come out of mitosis, so it's got to kind of reestablish itself. So usually when cells just finish mitosis, they're relatively small, so they got to build up their cellular components. They got to, maybe they need some more rough ER. They didn't get enough rough ER out of that division. They got to build up their organelles. They got to start building back up their lysosomes and creating more stuff for themselves to exist. So G1, get themselves set up, and then just a normal metabolic processes. If they are a pancreatic cell producing insulin, they'll just start doing that, right? So whatever that cell does, it's just gonna get itself set up and then just start doing that during the G1 phase, right? If it's a stomach cell that's pumping out pepsin and hydrochloric acid, G1 phase and interphase in general is where it happens, okay? So usually a lot of this time, especially early on, is spent building up ATP stores, reestablishing itself, building up parts of its components, its organelles, its lysosomes, whatever, so that way it can carry out its normal role for that organism. If this cell starts to get signals that, hey, you might divide soon, it will shift into this S phase, this synthesis phase. We're still in interphase though. So after G1, the next phase that could occur, if the correct signals are received, would be the S phase. And this is where DNA replication happens. But if it does not get that signal that you're going to replicate soon, that you're going to go through mitosis soon, if that signal is not received, the cell will enter what's called G0 phase. So we don't need this cell to divide right now. We just need it to stay put and keep doing what it's doing. We basically trigger it to go into what's called G0. It's kind of like pressing pause on the cell cycle. The cell is still alive. It's still doing all of its stuff, but it's not dividing. So we have some cells in our body that don't regularly divide. So our cardiac muscle cells don't often divide. Our bone cells are pretty slow at dividing, right? There's not a lot of cell division going on in certain cells in our body. Those cells are generally hanging out in G0 phase. So all of the cell's metabolisms continue. They're building up and breaking down molecules, creating ATP, processing molecules, right? Whatever it is that cell does, it's still happening in G0. It's just not moving forward, progressing with its cycle to prepare for mitosis. Because really S and G2 phases are preparation phases for mitosis. But we can come back out of G0. So let's say this cell does get the signals that it needs to divide and replicate. It can come back out of G0, okay? Once you go into G0, you're not stuck there. It's not like purgatory. You can come back out, right? If the correct signals with the correct hormones are received. So let's say our little cell does get those hormones, does get that signal that we need it to divide soon, that would shift it from G0 into S phase. And the S phase is where DNA replication happens. This is when it's happening in a cell's life cycle. So that process we talked in the previous video, DNA helicase, DNA polymerase, primase, all that stuff, this is when it happens in a cell's life cycle, during the S phase of interphase. S stands for synthesis. And what are we synthesizing? We're synthesizing DNA. We're building new DNA, right? So we talked about DNA helicase opens up, we end up with 2 new DNA molecules. What that looks like before the S phase, our DNA is in one single strand, okay? And we have, remember, we have 2 homologous chromosomes. These homologous chromosomes contain the same basic type of information, but different forms of that information potentially. And they come from different sources, right? Let's say this one maybe come from your mom, this one came from your dad. After that DNA has been replicated, this is what DNA looks like. See how now it's in this X shape, but they're still attached? This is one strand of DNA. This is the other strand of DNA. Remember how DNA replication, we end up with 2 molecules of DNA? Here's where they are. There's 1 molecule, there's 2 molecules. Okay? So we still call these a chromosome. While those 2, even though we have double the amount of DNA here, while those strands of DNA are attached at this middle region right here, we still call them 1 chromosome. Even though this molecule has half the DNA that this one does, even though we have this extra piece of DNA over here compared to before replication, we still call it 1 chromosome while it is attached at the centromere. This is important. This helps to figure out how we count chromosomes during mitosis. Okay, there's a lot of vocab words in this section that all sound very similar. So let's break them down. So remember, a chromosome is just a linear piece of DNA. Homologous chromosomes, one comes from mom, one comes from dad. They contain the same type of genes, but potentially different alleles. Okay? Each chromosome has a middle region where it's kind of constricted a little bit. It's called a centromere. Centro meaning center, and mere in reference to the chromosome, basically. So middle of the chromosome, that's what that means. After DNA replication, while those 2 pieces of DNA are still attached, and we count the number of chromosomes by counting the number of centromeres. So as long as there's only 1 centromere, and these 2 chromosomes remain attached at that centromere, we're going to count them as 1 chromosome. We call the pieces of DNA, though, when it's in this form, each replicated piece of DNA, we call it something specific. We use the term sister chromatid. While that replicated DNA is still attached to the other piece of DNA, we call each a sister chromatid. Basically, what mitosis is going to do, the process of cell division, is we will separate those sister chromatids to get back to this state of DNA. So each cell will get an entire set of DNA. Whew. Okay, so that whole process of DNA replication that we just talked about in that previous video happens in the S phase. That's the big thing we need to know about S phase. So during the S phase, there's going to be checks to make sure the DNA replicated properly, and there's no errors in the DNA. If any sort of error is caught in that DNA replication, and if the machinery to divide is not ready to go, we press pause on that cell cycle, because we don't want a cell to divide if its DNA is messed up. We don't want that messed up DNA to get sent on to another cell. That is how cancer is caused, among other things. But the idea being, there are mechanisms in the cell that kind of act like gateways, or like those little tolls you drive up on a toll road and you got to pay your toll to get through, right? There's basically enzymatic cellular processes that keep the cell from progressing to the next stage if something is wrong. So they'll put on the brakes, say, nope, you can't pass this stage, something's wrong with you, your DNA is all messed up, or something's going on, this cell's not ready to divide. If those problems are fixed, let's say DNA was not replicated properly, that problem can be fixed, and then it can go on to the next stage. Okay, so there are checks along the way to keep the DNA, to keep the cell from dividing if it's not ready to divide. If there's something damaged in the cell, or something isn't quite ready, something's not quite set up yet, that cell should not progress until it's ready, until everything is perfect. If that cell progresses, even if there's damage to the DNA, even if there's something wrong with the cellular mechanisms for replication, all the organelles that are involved with this, all the enzymes that are involved, if that cell blows past those checks, that's basically how we get cancer. So DNA is replicated during the S phase. The other thing that happens during the S phase, at least in animal cells, is our centrosome wakes up. Remember we talked about centrosome briefly, we just kind of waved our hands and said it's involved in cellular replication. We'll talk about it later. So the centrosome is an organelle that controls components of the cytoskeleton, filaments and fibers that will move DNA around during mitosis. S phase, DNA is replicated. The other thing that happens is we replicate the centrosome. So we get two of them now. Okay, we're going to have two centrosomes because we're going to need one on either side of the cell to move that DNA around properly. So those are two things that are replicated during S phase, DNA and the centrosome. So if any of this doesn't happen properly, cell puts the brakes on and says, nope, go back, try again. Let's get this right before we move on. So DNA is replicated, centrosomes replicate, everything's good to go, we get the green light, we can move on to G2 phase. So G2 is like getting ready to move. You're gonna maybe save up some money so you have enough money to pay for a moving truck or whatever. You're gonna compact all of your DNA, you're gonna make sure you have everything that you need with you, right? So G2 phase is a preparatory phase. G2, we call it the growth 2 phase. We're making sure we have enough ATP to proceed. We're making sure that the proteins that we need to move DNA around during mitosis, that they're there, that they're present. Because if those proteins aren't there and we start replicating, or we start mitosis, that's not good. Mitosis can't happen if those proteins are not assembled properly. We actually start to break down the cytoskeleton because we need the organelles to be able to move around freely. So we're going to start to move the organelles, everything around the cell is going to get kind of shuffled up. So that way we get two cells with an exact copy of everything. So we also start to build up the organelles too, because this one cell will divide into two identical cells, and each of those cells is going to need a Golgi. It's going to need some mitochondria. It's going to need some Repi-R, some Smoothie-R, some peroxisomes, some lysosomes. So we build up the number of organelles in our cells during G2 phase. So that way each cell, once it finishes mitosis and starts into G1, it will have enough cellular components to get started with its metabolic processes. And then we have another set of checks. Is everything okay? Do we have everything assembled? Are we good to go? Do we have all of our centrosomes? Is everything ready? There's another check here, making sure that this cell is prepared to divide. If we don't have enough organelles, if we don't have enough ATP, if DNA is not replicated properly, if anything is wrong, we put the brakes on and go back and try and fix it before we move on. And again, if those checks are not in place, that can cause cancer. If a cell replicates before being ready, that can cause cancer. So let's say we do get the checks, we're good to go, barrier lifts up, all right, let's head into mitosis. And that's where we'll pick up in our next video.